So, welcome everybody. Thanks for coming. Despite of the weather, some people are here, and we also welcome our guests online. So, it's my pleasure to welcome Professor Tobias Erb, who is here this evening to deliver the fourth lecture of our Frontiers in Science series. With Frontiers in Science, we want to take the current developments within the field of international cutting-edge research and convey them to a broad, multidisciplinary audience within our university public. Each semester, a renowned researcher joins us to discuss their work and its implications for the 21st century challenges in science, politics, and society. Together, we explore leading questions, theories, and methods pushing scientific boundaries. No doubt, climate change is a pervasive issue that affects every corner of our planet, from rising temperatures to extreme weather events. The impact of a warming world are undeniable and far-reaching. The challenge demands our immediate attention and concerted efforts to mitigate its effects and adapt to its inevitable changes. Addressing climate change requires innovative and robust solutions, with technology playing a crucial role. Among the many technological approaches to mitigating a changing climate, synthetic biology stands out as particularly promising. By engineering biological systems, synthetic biology offers novel ways to produce biofuels, create sustainable materials, and even develop new methods for carbon sequestration, and we will hear about this in a minute. With Tobias Erb, we are honored to host an acclaimed researcher who is at the forefront of this exciting field. Tobias Erb is a director at the Max Planck Institute for Terrestrial Microbiology, as well as professor at the Philipps University, both located in Marburg. His research group interfaces biology and chemistry and centers on the discovery, function, and engineering of CO2 converting enzymes and pathways. Research in Tobias Erb's lab crosses multiple scales, from the molecular mechanisms of carboxylases to their ecological relevance, and from understanding the evolution of natural CO2 fixation to developing new-to-nature solutions, something he'll talk about in just a moment. Tobias Erb was awarded a PhD in 2009 at the University of Freiburg and the Ohio State University. After a postdoctoral stay at the University of Illinois, he headed a junior research group at ETH Zurich before relocating to the Max Planck Institute in Marburg, where he was promoted director in 2017. Tobias Erb has received numerous awards, among them the research awards of the Swiss and the German Societies of Microbiology and the Future Insight Award in 2023. He was named one of the 12 up-and-coming scientists by the American Chemical Society in 2015 and elected to the European Academy of Microbiology in 2019, and to the European Molecular Biology Organization in 2021. Last year, he was elected to the National Academy of Sciences, Leopoldina, and this year, he has made the honor to being awarded the Gottfried Wilhelm Leibniz Prize of the German Research Foundation. Dear Professor Erb, we are delighted to welcome you to TU Dresden. And before we start with your exciting talk, I would like to hand over to Professor Thorsten Mascher, who holds the chair of General Microbiology at TU Dresden, and who will briefly introduce Tobias Erb's field of research to us and the role it plays at TU Dresden.
Yeah, so welcome also from my side, Tobias. When I was asked to give these few words of introduction, I was happy and pleased to do so. We know each other for some while. And so my job is to introduce the field that he's working on, and it's already in the title. It says SynBio, synthetic biology is part of it, so that's the realm we're about to hear. And that is really maybe kind of the latest addition to the many fields of life sciences, but it started in about 2004, so 20 years of synthetic biology already in. And for those that are not familiar, I think most people are, it's not a contradiction to say synthetic and biology. It really is more in the realm like in chemistry, where you have analytical versus synthetic chemistry. So it's applying what you learned in analytical sciences to design new functionalities. And that's what synthetic biology is, to design, implement new functionalities in living systems, or to redesign existing natural systems for useful purposes. So it's an engineering discipline. It brings together the knowledge from chemistry, from biology, and the approaches from engineering. And it comes in many, many different flavors. So one of the big toy fields in the beginning was logic gates, circuitry. People did this in the first decade, I would say, of this century. And this is kind of a tick off. So all the logic gates, your AND gate, your OR, your NOR gate can be implemented into cells. And bottom-up genomics was the next big thing. It's still ongoing. And big questions are linked to that. What is the minimal functions of life? What are the minimal gene sets in life? That's what Greg Venter was doing. Or how to make a better E. coli, or how to make a better yeast. That's ongoing projects. Or bottom-up minimal systems. In fact, that's something that is going on pretty strong here at TU Dresden. We had Petra Schwille a couple of years back. She was doing that. She relocated to Munich many years ago. But with Dora Tang, for example, or James Sunch, there are people that do synthetic systems in lipid droplets on lipid membranes to set up minimal systems that simulate life or help to rebuild living systems. But there is also here metabolic engineering, and that's in fact the field that is much closer to what Tobias is doing. So Thomas Walter, for example, in bioprocess engineering, he's heading the group of synthetic biotechnology and systems biology. And metabolic engineering, or the synthetic biology metabolic engineering realm, is really trying to bring together the knowledge that we have generated in the last 20, 30 years and try to implement it for useful purposes. That could be biofuels, that could be pharmaceutical compounds. Artemisinic acid is one example from Jay Kiesling, for example. But I think if you would name two fields in metabolic engineering, the SynBio way, as the holy grails of synthetic biology nowadays, it's either implementing nitrogen fixation into living systems or moving this, or to use and apply photosynthesis and carbon fixation and move it into new hosts to improve that. So these are, I think, the two big goals that many groups work on. And I think when it comes to carbon fixation, Tobias Erb is most probably one of the leading figures worldwide, and definitely the leading figure in Germany on that field. Try to apply what nature has evolved, try to improve what nature has evolved, first in bacteria, later in plants, for dealing with big problems like climate change, like dealing with carbon dioxide emission and trying to fix that. And without further ado, Tobias, this is what you're going to talk about. You can do it much better than I can. So I leave the stage up to you. I'm very happy that you're here. And I think we can look forward to a very thrilling talk in the realm of metabolic engineering the SynBio way. Thanks for coming.
And thank you so much for your applause. Please don't applause, you don't know what to expect, so be a bit more careful. And when I was contacted to give this lecture, I felt very pleased and also privileged, because it's one of the rare occasions where you can, of course, talk about science, but also develop a little bit more of a vision where science could go in the future. And just as a small spoiler, I'm not going to save the world from climate change today, probably also not in my lifetime, but I could give you a vision where we could move together as a community, as scientists, maybe as a society, and also how the different fields of research can contribute to this big challenge and what new can actually be created when these different fields meet each other in an interdisciplinary fashion. And so this is why I named my talk Breaking the Limits of Natural Photosynthesis with SynBio. And you might realize that I also put a question mark at the end, which is different from my original title, which was a statement. This is a question, because it's still open to me if SynBio can deliver at that scale, but I guess we should at least try it, and there's also reasons why we should try it. All right, so all at the end is about a future. I would like to take you with me. But before we go into the future, I'll take a step back and look a bit back in the past of this planet, or on the planet, its current stage. And this is just a reminder that this planet is a living ecosystem, and there's many different species, billions of different life forms that inhabit this planet, right? But it's also important to note that one species is really actively shaping this planet. And that's very important, and this is clear if you, for instance, look, then you know what kind of species I'm referring to. It's human beings, it's us as humans on this planet. There's just one figure that shows you how much we actually influence this planet. This shows you actually the accumulated anthropogenic mass, so the mass that was created by mankind over the last years, and we have hit around about 2 or 3 years ago the point where we have created more mass as human beings than there's biomass on this planet. And this is very interesting to me, just as a keep in mind as for the reminder for the rest of the talk. There's just one figure, but there's much more, and it actually shows quite dramatically how, let's say, since the 1950s, we have started to really shape this planet in very different ways. So what I show you here is, for instance, how the population is taking off over the last decades, right? And this, of course, comes, for instance, with an increased water use. This comes, of course, with increased transportation. So all of this is, let's say, social trends, societal trends. And at the same time, there is also consequences, right? And you can see these consequences. So what you could see here is, for instance, how much loss of biodiversity is because we need more space as human beings, right? You see how much oceans are acidified due to climate change. You see how the surface temperature increases. And there is probably 20, 30 different factors that tell you how human activities influence this ecosystem dramatically. And this is called the Great Acceleration. The Great Acceleration is that we actually really start to really influence and dramatically change Earth at an unprecedented pace. And probably the most important, most relevant molecule that we are interested in that changes the planet dramatically is this small molecule here called carbon dioxide, or CO2. And I think we all agree that this is a very potent greenhouse gas, and I think we also all know that over the last years, decades, actually centuries, the amount of CO2 has also been increasing steadily. And this is known from the famous Keeling curve. That's basically recorded yearly how much CO2 is in the atmosphere. And just to let you know, this is not much. It's actually 420 parts per million. This is around about 0.04%. So in this room here, it's probably a couple of bathtubs of CO2 that are finely dispersed, that are hanging around in the atmosphere. And we all know that these concentrations have increased, and we all know that this increase of concentration of CO2 have direct impact on climate change because they lead to this greenhouse gas effect, which is amplifying. And it's typically the way we look at CO2, being a greenhouse gas, being a threat to society, to the planet in general. But today I would like to turn your view around, and I would tell you CO2 can also be a chance. Why? Well, in principle, it's a simple carbon source. It's a carbon source that sits there in the atmosphere, and in principle, we could and should be able to create all our materials that we need, all our food, all our, you can see a lot of chairs made of wood. In principle, we should be able to capture the CO2 from the atmosphere in a sustainable fashion and convert it into useful products. And this would, of course, close the global carbon cycle. This would immediately lead to a balanced interaction of us human beings with the planet. Well, this sounds like a great plan, and I think we as society, as scientists, should be able to do that, but the point is, we are really bad at it, at least in physical chemical terms. So there is no single physical chemical process, no single catalyst that would allow us to capture CO2 at a global scale in a sustainable fashion and produce products of our everyday use. And in fact, all our chemical technical efforts are still largely outcompeted by biology. And this is sustainable CO2 physics you will see here. That's the green jungle. This is basically light-driven carbon capture in a sustainable way. So I'm telling you is that plants are very efficient CO2 filters. And we actually use this technology already for centuries, even for more than thousands of years. And this is just a picture back from Marburg, where I'm currently located at. This is the Amöneburger Becken, where you see that we actually can use plants to fix carbon dioxide. Agriculture, right? So we can actually make use of this fantastic blueprint which operates at the global scale. So it's fantastic. So there's a solution to fix CO2 with light, converting into biomass. We can use this technology, and we typically think of plants as being really important figures on this planet. That's true, but what's also true is that there is also microorganisms. And I'm happy that Torst is sitting here because he loves microorganisms, so do I. What sometimes gets forgotten is around about 30, maybe even 40% of the carbon dioxide that's fixed on this planet is fixed by these small microorganisms. Okay? You just don't see it because it happens very often in the soil, and it happens even more often in the oceans. And you need a microscope to see CO2 fixation. By the way, microbes invented CO2 fixation. The chloroplast is nothing else than a form of cyanobacterium. So if you want to really talk to the experts, those are the experts. And I'll show you one expert here. It's called Prochlorococcus or Synechococcus. So those guys fix around about 50 gigatons carbon dioxide per year. And they produce, of course, a lot of oxygen. And there's calculations out there that every, let's say, 5th or 6th molecule of oxygen you breathe in this room here is produced by these microorganisms. So this tells you about the relevance of these small players in the large global carbon cycle. Well, there's apparently a solution. It's called photosynthesis, and so we could say the problem is fixed. Well, it's not that easy because photosynthesis is sustainable. Yes, it can do CO2 fixation, and it actually does it at about 400 gigatons of CO2 per year. But the problem is that natural photosynthesis is not perfect and not sufficient for us human beings. This becomes clear if you look, for instance, at the way how we can capture CO2 through agriculture and forestry. So there's around about 10 billion tons of carbon, which is around about 40 billion tons of CO2 that we can capture and convert annually through agriculture in a sustainable fashion. That's around about now we can do as human beings with this old school technology, agriculture, forestry. Well, at the same time, we need more energy, and we actually create this through releasing additional amounts of CO2. And we see it on the top of it. We emit around about 10 billion CO2 per year from fossil fuels. So the global carbon cycle is imbalanced. And you can do the math. If you want to really now balance this global carbon cycle, you have a growing world population, you have more energy demands. If you just would use natural photosynthesis to try to bring the cycle back into shape, you would need probably 2 to 3 Earths until 2050. All right. So natural photosynthesis is there, but it is not running at a pace that we would need. And you could ask, what limits actually natural photosynthesis? So what is the limiting factor in CO2 fixation through plants or photosynthetic microorganisms? And it's a long-standing question, and it boils all down at the end of the day to the actual enzyme, the biocatalyst, the engine that drives carbon dioxide fixation in the plant. This is called ribulose 1,5-bisphosphate oxygenase carboxylase, or short Rubisco. And that is the catalyst that grabs CO2 from the atmosphere and puts it into the metabolism of the plant. I actually brought it here with me. So for each person in this room, there's 5 kilograms of Rubisco. So this is probably only 800 grams. But it tells you how important and how relevant this enzyme is. Take a look at it. It's everything you ate today was fixed to this enzyme. And it's a fantastic catalyst because it can grab atmospheric CO2, but it's actually also lousy because it's very slow. So an average Rubisco takes probably 5 to 10 CO2 molecules per second, which is surprisingly slow compared to other enzymes we know. So it's slow. And there's another problem. The enzyme is also sloppy. It can actually not only fix CO2, it can also take oxygen from the atmosphere. And that is really a downer, right? Because if this happens, there's a process called photorespiration which takes part, and the plant actually starts to release CO2 that is fixed before. And this error is substantial. So Rubisco has an error rate of around about 20%, which means every 5th time, instead of fixing a CO2, Rubisco picks up an oxygen molecule, and this is dramatic, as I just told you. So you could say the engine of photosynthesis is very slow, and it also stutters. Now you could ask, hey, we have SynBio, we have protein engineering. Can you make the enzyme faster? And in fact, you can make the enzyme faster. But then you automatically make the enzyme worse in terms of distinguishing oxygen from CO2. You can try to do the opposite. You can try to make the enzyme more specific, but then automatically catalysis slows down. What I'm telling you is that Rubisco is trapped in a so-called, scientifically speaking, Pareto optimum, where you cannot touch one parameter without negatively affecting the other one. And you should think about catalysis really as these two things, like speed and specificity. And a good example might actually be you typing into a mobile phone. You either can type very quickly, but it automatically do a lot of mistakes, and the message will be really bad to read. Or you can try to type very accurately, but then you are also very slow. And this is exactly what limits catalysis, at least in the case of Rubisco. It hits a so-called Pareto optimum. And this has been really the starting point for our research. When we really figured it's really hard to improve this catalyst, why don't we take a different step? Why don't we just let Rubisco be Rubisco, photosynthesis be photosynthesis, and think about other ways, new ways, different ways, complete Rubisco-independent ways to fix carbon dioxide, a process we would call synthetic CO2 fixation, and build those pathways from scratch and hopefully hit a new optimum. Now you could ask, isn't it too bold? It's billions of years of photosynthetic evolution. Can you actually reinvent and find new ways to fix carbon dioxide? And you might want to be humble and actually take a lesson from nature. What I'm showing you here is the space, or the biochemical solution space that nature has conquered already during the billions of years evolution. And what you might not know is that besides photosynthesis or photosynthetic CO2 fixation, nature has actually already found other ways of CO2 fixation. Surprisingly, or not surprisingly to me, a lot in different microorganisms. And what you see here is that nature actually has already found 6 different solutions next to Rubisco. And that's the solution space that nature has conquered. And as I said, some of them we already use. So I just showed you photosynthesis on the lights and forestry, agriculture. And some of the microbial pathways we actually also can use already at large scale. For instance, here shown these acido and methanogens, which we can use, for instance, to convert CO, carbon monoxide, hydrogen, and CO2 to high concentrations of steel mills. So nature offers different solutions. That's fantastic. So me as a biologist, I'm looking at this landscape, and I think it's really cool how creative nature has been. But me as a synthetic biologist, I look at the very same landscape, and I think, there's still a lot of space. There's a little void, and a lot of things that nature has apparently not discovered or touched, or maybe just not figured out. And this is what we want to do. We actually want to go into this empty space and we'll build new solutions, new islands in this vast ocean of possibilities, and create those new solutions from scratch. So it's not metabolic engineering in a sense that you try to improve something existing. It's really trying to build something fundamentally new. To give another example or another picture, think about how we humans learn to fly. We actually studied birds. The biological example of flight, we extracted the fundamental principles, and then we built a plane. But the plane we built does not flap its wings, neither does it have, nor does it have feathers, but it flies. And so that's exactly what we're trying to do. We try to extract the fundamental principles of how nature captures CO2, converts it into biomass, and how it's powered by light. And then we want to build a human-made solution that is as efficient and does exactly the same thing, but it might look very, very differently. So how are we going to do this? In our lab, we have basically laid away a workflow towards this synthetic CO2 fixation. This starts all off with just design. Thinking about how an optimal CO2 fixation pathway, an optimal photosynthesis, an optimal operation system of photosynthesis would look like. We evaluate the different designs. We then go ahead. Once we have a good design, we try to find the parts. And parts are, biologically speaking, enzymes, proteins, biocatalysts. We put the system together, try if it runs, try to optimize it in different rounds. And once we have really robustly operating system, we would like to move it inside of a cell, and this could be a natural cell or an artificial cell, and I will talk about both of it later in my talk. And of course, at some point, we actually might want to go away from single cells, microbes, or photosynthetic microorganisms, to higher organisms, and the big goal will, of course, be also to touch the chloroplast in the plant at the end of the day. All right, so how do you design these new cycles? The first step, how do you think about photosynthesis really radical and new? What you do is a principle that I have learned as a chemist, because I'm a chemist and a biologist. It's called metabolic retrosynthesis. So you really sit down and you start to think and draw potential transformations of metabolites. And these are a couple of cycles we've drawn, a couple of cycles we've already built in the lab, and all of these cycles feature very different reactions. Important to note is just theory. could just draw it here on this blackboard. That's actually what we do. We sometimes draw these cycles, and then we think about those cycles, which of them is a good design or not. Don't memorize the different chemicals in there, because all of these cycles are very different in terms of topology and chemistry. But they also have a couple of common principles. And one principle is that all of these cycles are, as I said, cycles. So these pathways are cyclic, which means they can go in rounds and rounds and rounds, and it can continuously fix CO2. That's one principle, so a cycle to continuously fix CO2. And the second principle is that all of these cycles are so-called split reactions, reactions where we can take off some of the fixed carbon and convert it into a building block of life, or a building block of chemistry. Now, there's different solutions. I showed you Ketch cycle, Hopek cycle, theta cycle, probably 10 or 20 more cycles we've drawn. The question is, which of these cycles is actually a good solution? So how do you evaluate your designs? And this comes back to chemistry. So what we do is we basically have different simple parameters where we look at what would be helpful for a good design. And one, of course, important parameter is kinetics. In other words, speed. So the question, how fast can you turn such a cycle? And I'll give you one example for the Ketch cycle. This Ketch cycle is basically not based on Rubisco, as I said before, because it's a really slow catalyst. This Ketch cycle is based on a new principle of CO2 fixation we discovered around about 15 years ago in microorganisms. It's called NO-ECO-A reductases, ECRs. And those are highly efficient CO2 fixing enzymes. And you can actually see them. That's a simulation we've done with partners in Chile, how good they are in capturing the CO2 from the atmosphere and making a CC bond directly. So that's the active site of this catalyst. And they're actually 10 to 20 times faster than Rubisco, which means at the same time, you can fix 10 to 20 times more CO2, which is an increased space-time yield. So kinetics matter, speed matters, but what also matters is thermodynamics, or in other words, energetic efficiency. You want to drive the Porsche fast, but you also don't want to use a lot of gasoline, right? So exactly that's what we're also looking at. So how much ATP, how much NADPH do you need to actually operate these cycles? The Ketch cycle is a good design because the CO2 fixation should be 10 to 20 times faster, and it only needs half of the ATP that you would need as a natural photosynthesis. So this looks like a really great design. Now comes the more important part and the more exciting and the more challenging part, which is you need to go back to the lab and you want to build this system. So the evaluation, design takes you probably 1 to 2 weeks. The actual challenge is to find the individual parts, the enzymes, the proteins, to build the Ketch cycle. Where do you find these parts? There's not a shop where I can simply drill it or I can simply make it, and I think we are far away from designing enzymes from scratch, although we have started to do that as a scientific community. But what we typically turn to our attention is microorganisms, because they are just so diverse in terms of biochemistry, and they are so different that you would probably find for each enzyme that you intend to build and to use in your cycle an equivalent in nature. Just to give you one reference, if you have 1 gram of soil, and on this spoon it's probably 7 to 8 grams of soil, you would have 1 billion of these microbes. That's a fantastic diversity. Look at this spoon. This is basically the whole world population on 1 spoon. And there is different species in there. And this is a picture that shows you how diverse these microorganisms are. They come in very different flavors. And every microbe has around about, in average, probably 4,000 genes. We just know half of their functions. And each microbe also has unique genes. So this biodiversity is just mind-blowing to me. And this is what we're trying to do. We just screen for the individual parts that we need to build a system through these microbes. Or we talk to colleagues like, for instance, Thorsten, Mascha, or for instance to Michael Rother, who have all these things explored in their favorite microorganisms, and we sometimes take the parts from them. To make a long story short, so we had this challenge of finding enzymes to build the first version of the Ketch cycle, and we actually found those in very different organisms. For instance, in Rhodobacter, it's a classical photosynthetic microorganism that is in the lab. We just sneaked and stole a couple of genes or proteins from. E. coli, probably also well known as a gut bacterium, we took 1 gene from. We actually used genes from Methylobacterium. It's an organism we find on the plant surface. Each plant has Methylobacterium on its top, on its leaves. Nitrosopumilus, which is an archaeon, different domain of life. We also took some genes. We actually had a gene from the human liver. It's not a microorganism, I know. And we also have even genes from Clostridia, which is very close to the bacilli that Thorsten is working with. And we even engineered and took one enzyme from Arabidopsis thaliana, a small plant, that we re-engineered to do the reaction we wanted it to do. So it's a patchwork pathway, but we put all these enzymes together in one Eppendorf cup. We actually added ATP and NADPH, the energy, chemical energy, and we could show that the cycle really starts off turning and it starts to convert carbon dioxide into malate, malic acid, which is a fantastic building block for cells, but also for chemical industry. So what I was telling you is that we really can think of a new operating system, a complete new pathway for CO2 fixation. We can go in the lab, we can build the pathway, and the pathway operates. So this is synthetic biology. And we were really super happy to have built this first version until we looked at the kinetics, at the speed, and we actually realized this cycle is awfully bad, and it turns. And this is something we've faced now several times by building different solutions. We very often build something that is somehow functioning, somehow doing its job, but it's far away from the optimal. It's very slow, it falls apart, it's scrappy. It's almost like you start to climb a mountain, you're at the base camp, but you still have way to go to hit the top. And this is exactly what we have to do typically in the lab when we do SynBio. We have to optimize, to improve, to build a second, a third, a fourth, a fifth version. But why have you to do that? So why is the system not better? Well, the point is that all of these enzymes are put together from very different biological backgrounds. They don't have a common history. And very often things go wrong and the cycle stops because one enzyme steals a substrate of the other enzyme. One enzyme is not specific and branches off a metabolite should actually turn in the cycle. Or you just have side and waste products that fall off the cycle. So what I'm telling you is, even though you think about all the individual enzymes as individual players, you try to put a really nice team together, the team is not as good as you think. And just because we have European championships, I brought a good example. So, you know, it's not just about thinking each individual player as getting the best person in the team. You really need to think about the team being the best team of players. What I'm telling you is it's not an optimization function on the individual parts, but of the whole network, of the whole system. It's a different challenge. So what can we do? How can we improve the system? Well, in principle, you can train some enzymes to become better. You can train some enzymes to do the right reaction and ignore the other reaction to hit the goal better. That's one possibility, and it's actually what we did. We engineered and redesigned certain enzymes to become better in doing one or the other reaction. You can substitute enzymes, right? You can just swap in or swap out enzymes so that you get a better playing team together. And in fact, we actually have detoured one reaction. You see it here. Because the original enzyme wasn't good, we substituted it, right? So that's also possible. And then a team is more than its players. You also can actually put in more enzymes, supporting stuff that keep primary metabolism up and running. And I think that's a principle that is very different to a chemical physical audience, where you build a perfect digital part. Biology is messy. It will always make mistakes. And biological systems are robust because of additional reactions ongoing. In your cells, you have primary metabolism, but you also have, of course, a lot of cleanup enzymes that remove toxic metabolites, that detoxify things. And if you want to build a robust system, we actually make it robust by adding such additional supporting helper enzymes. And I think that's a fundamental difference in building synthetic systems compared to chemical and physical systems. Okay, if you implement all of these things, you change also the turf a little bit, you actually see that can improve the catch cycle in its operation by almost 20-fold without changing its topology or its basic chemistry. So you go through all these versions, version 5.4, it's already very good. It's much better than the original version. It's at the stage where you are if you take a plant leaf, you break the plant leaf, and you try to measure CO2 fixation. So this is probably really where we are. This slide I'm showing you here took us probably 2 to 3 years. So that's just summarizing the work you have to put in to optimize such a system. And I'm really glad that Thomas Schwander, the first author, took so much energy to try to optimize. And I guess you can also appreciate it's so complex a system, sometimes you don't understand what's ongoing and it's too complicated to oversee all the interactions. And this is what has inspired us the last 2 or 3 years to think about, can we not go a different way and systematically explore the optimum, use other ways? And there it comes into play that it's important to also have technologies at hand. And one thing we've started to do is now pipetting and putting different versions of the cycle together where systematically vary the content, the concentration of the individual enzymes. And we actually started to go away from the manual labor in the work, in the lab. So no cell-based manual work anymore. We actually start to use automation and miniaturization. So for instance, we have this pipetting robot here, which uses ultrasonic sound to move liquids just across, no pipette tips, no plastic waste, femto picoliters that are just moved from A to B with ultrasound, contact-free, super quick, and probably more error-free than a PhD candidate in our lab. I'm sorry to say so. The point is, this opens up, of course, parallelization, and allows you to do a lot of different variants of the cycle. So you can work in 10 microliters now, 384 wells in parallel, maybe 1,500 wells, and you can do a lot of more assays. You can analyze those assays, and you can feed it back into a learning algorithm that tries to predict again which of these parameters you've changed are important and suggests a new round of experiments. If you apply this design, build, test, learn cycle, it's a typical thing you do in SynBio, you actually see that within 8 rounds of optimization, you shoot up by a factor of 60 compared to the first version you have built. And this took us only half a year. So this gives you roughly an idea how fast you can actually speed up if you can generate larger data and if you actually have the help, for instance, through machine learning algorithms. Now, where are we now? Well, where stand we, for instance, compared to chemistry? So if you think about chemistry as general, as a field, you typically start off with crude oil, you start off from fossil fuels as a resource to do chemistries, to do transformations. And chemists typically can do 1 or 2 transformations parallel, then you have to support to extract your product, move into a different reaction vessel, do the next reaction. In a stepwise process, you can build up your molecule. Now, the catch cycle is actually different in the sense that it's 10 times better, or maybe 20 times better, because you can already operate 10 to 20 reactions in parallel. And you can start off CO2 to make a small organic acid, in this case called glyoxylate. You can also make malate. We can adopt a little bit what the outcome is. But overall, you basically have now a system that can do many more reactions in parallel, a factor of 10 more than a chemist would be able to do, and it can start off CO2. Now, can we convert this glyoxylate in more interesting compounds? Can you, for instance, make antibiotic precursors, amino acids, or food flavors from it? In fact, you can do that. We've done this already. And just one slide. I don't want to dive into the details. Just want to give you one example how we were actually able to produce the antibiotic precursor erythromycin, or more concrete, 6-DEP, which is the precursor without the sugar moiety attached, by coupling more than 50 reactions to each other. And it's not only coupling the reactions. Again, you have to optimize, you have to finely tune the enzymes, and you have to design new pathways to make all this happening. But in principle, it's possible. So I told you already how good we are, and of course we are proud of it, right? And we also are really proud that the system has started to work after 10 years of let's say, laboratory evolution and human design. But of course, we are still far away from what cells can deliver. If you think about cells like Torsten is working with, right, living cells, they can operate 1,000, maybe 2,000 different reactions in parallel. That is just amazing to me, how you can control in time and space all these different reactivities. And so this inspired us to think even beyond this 50 reaction, think about more in the context of a living cell. There's also something you're doing in Dresden, for instance, the physics of life, where you start to extract those principles, how life operates. And it's more than just chemistry. Think about the chloroplast or photosynthetic microorganism. It is light-powered, so it can convert energy that comes from the outside. It can actually do its metabolism to sustain and to grow and to divide, and it can self-replicate, and it can self-optimize. So those are all the functionalities that such a system offers. And it's far away from these small enzymes that we have in our Eppendorf cup. And so the idea would be, can we actually now also power the system instead of providing ATP and DPH? Can we use light or other sources of energy? Can we structure it in 3 dimensions? Can we eventually also put information in there? Can we actually get a self-sustaining system? And these are all the things we are currently exploring in our lab. So maybe the first question, can we simply power the system with light? So can we take the synthetic catch cycle we have designed in the lab, and can we just couple it to the native photosynthetic machinery? So can we, for instance, take spinach that we buy in Marburg on the market? Can we grind up the spinach, extract the photosynthetic membranes, and shine light on the photosynthetic membranes, and use the ATP and DPH to actually power this synthetic catch cycle? And interestingly, this is not possible, right? That's very interesting to us. So what we figured is that the catch cycle, synthetic design, and the natural energy machinery of the photosynthetic cell cannot talk to each other. What you have to do is you have to go back and find the bottlenecks, the roadblocks. You have to swap out 1 or 2 enzymes, and only then those 2 systems operate with each other. What I'm telling you is that even in cells, the individual modules energy regeneration, metabolism, do not evolve independently of each other. They are finely tuned and synchronized with each other. Stoichiometries play a role, or chemistry's toxicity. All right. Okay, so this is just about powering such system, but you can even think bigger and broader. Think about building eventually also an envelope, an artificial cell around this metabolic core, this metabolic heart, this metabolic operating system. So how do you do that? Well, if you think about cells, at the end of the day, it's nothing else than water droplets surrounded by an oil or membrane, and again, hanging around in water. It's nothing else than a vinaigrette, if you want to say so, right? So it's nothing else than small droplets, water in oil, in water droplets, or water in oil, mini droplets, that you can actually create if you, for instance, mix vinegar together with water. So of course we don't do that to make these small droplets. What we use is a technology called microfluidics, and this allows you to actually produce thousands of small droplets, water and oil droplets per second. And you can use these microdroplets as empty cells, if you want to say so, as cell envelopes, and you can start to load those envelopes with different technologies and techniques. And you can, for instance, put in photosynthetic membranes. You can, for instance, also put in the enzymes of the Krebs cycle. And then you end up at something that we would call an artificial chloroplast, a system that contains these photosynthetic membranes shown in green. It is like cell size, a bit bigger than a yeast. And if you shine light on this system, it actually starts to power up, and it starts to generate energy. And what you see in blue is NADPH. If you turn off the light, the NADPH can be used to fix carbon dioxide. So you can really charge, decharge, charge, decharge, charge, decharge the system roundabout for 2 hours. And you can fix CO2 and you control this activity in time and space just like a real chloroplast. All right. So I told you already a little bit how we now move away from this natural system towards a blended system where you have some natural parts, some synthetic parts, and we have this artificial chloroplast. It's not very stable, but it actually operates, and you can control it. It's like a small cell-like organelle. But of course, you can think bigger, especially at a place like here where you have also, let's say, hardwired physics or material sciences. You could think of moving up this ladder towards a bit more hardwired world. Think about where we can get energy from. We can, for instance, use electricity. And would it be actually possible to interface biochemistry with electricity? Would it be possible to power CO2 fixation directly with electricity? It is something we've also started to think about recently. So what you need to run the cycle is, on the one hand, redox reactions. You can directly use electrons. But you also need ATP. And ATP is the one compound in your body, in every living system, that provides the cell with energy. So if you move around, you need a lot of ATP. Every person in the room probably has 70 kilograms of ATP that is synthesized per day just by walking around and being a happy human being. So the question is, how can you generate ATP? And can you generate ATP in a different way than nature does it? Because nature does it in a very complicated fashion. It actually uses charge separation at membranes and many, many different complexes. So nature creates a chemoelectrical gradient across the membrane, and this gradient is harvested by ATP synthase, a rotating protein complex that then produces the ATP. I don't dare to build such a system. This is way too complicated for me, but I can think as a synthetic biologist again. I can think, is there other ways? Could we directly interface, for instance, proteins with electricity or with the technical electrical world? Can we use a minimal set of components, maybe no membranes at all? And can we actually use this as a core, an energy module, to power chemical reactions? And what we found is actually a way how to do that. And this is shown here. It's a protein called AOR. It's a, if you want to say so, an adapter between the biological and the chemical or technical electrical world. It is a protein which can accept electrons, low potential electrons. And the protein has iron sulfur clusters inside. And those iron sulfur clusters are like wires, nanowires that can accept electrons, and the electrons can move from the outside all the way to the reactive center of the protein. And this is where chemistry happens. And you can, for instance, reduce an acid to an aldehyde. That is pretty hard, typically for an enzyme, without activation. But if you have low potential electrons, this enzyme is exactly and very efficient at it. And what you can do now is you can basically put the enzyme on an electrode. You can feed it with electricity, it would reduce acids to aldehydes, and then you can close the cycle by just oxidizing again aldehydes to acids. And in this process, you can release ATP, and you can use this ATP, for instance, to make starch out of glucose, or starch precursors out of glucose. You can even power transcription translation systems in vitro with this system here. What I'm telling you is this is probably one of the most exciting discoveries and designs for me, that you really can use electricity to power and produce the molecule of life from the power molecule of the cell directly from electricity. All right. I talked a lot about artificial cells because that's also a strong part here in Dresden, this kind of bottom-up construction of minimal systems to some extent. But what about natural cells? So can we, for instance, take the whole operating system, and instead of building a complicated artificial cell around this metabolic operating system, can we, for instance, simply take this operating system and bring it back in the context of a natural cell? So can we take the catch, the theta, the HOPEC cycle for that matter, and can we boot this software inside of this billion-years-old hardware? And you might appreciate this is a big challenge because cells have billions of year evolution, and the question is, are they still able to learn new things? It's like as human beings, are you able to learn a new language? Are those cells able to learn a new operating system? And how are you going to do this? So what we do is we basically create incentives for the cell. We go into the cell metabolism, we look what the cell needs, and we actually create dependencies. Let's take a simple cell like E. coli that needs, for instance, a compound shown in yellow, which is called succinyl-CoA. That's a chemical compound, a metabolite that E. coli needs to make a cell wall or lysine, for instance. Now just block every pathway that synthesizes succinyl-CoA. Just stop metabolism from producing this compound. The cell is dead. It cannot grow anymore. That's a strong, or well, it's a killer, basically. It's just, if you knock out all these genes. So the cell is dead until you provide the cell with a new way to actually make this compound. And this is what we have been doing. We actually brought in parts of the catch cycle, not the full catch cycle, but parts of the reaction sequence. And it can actually produce now its succinyl-CoA through CO2 fixation, starting from crotonate. And the cell can start to grow again. And that's one of the first examples how you can actually put part of the catch cycle inside of a living cell, and it actually fixes CO2 and provides the cell with some biomass. Now, this is a starting point to do real evolution. It's also a starting point to not only make lysine, maybe, or the cell wall from CO2, but eventually make the whole biomass from CO2. And that's the way we actually want to use now in the future, living cells to optimize our systems and to get them onboarded into the background of natural cells. Okay. So what I've been showing you doesn't save the world from climate change. It's too small of droplets or too fragile or far away from being already fixing so much CO2 that you can actually make a difference to this planet. But we actually have started to think how to build such a system, and we have actually shown you can build new solutions to the space. And that's the motto of the Max Planck Society, which says insight must precede application. So aspiration is not necessary to have it in 2 or 3 years. We think just broader and more visionary, hopefully. And this also keeps us doing these things in the lab. If I have another 5 minutes, I would just try to also tell you these insights we've gained can tell us also about real-world solutions that we can create now. And for that, I want to come back to the initial slide where I talked about how bad photosynthesis is and about the process of photorespiration that is actually limiting Rubisco. And I told you about this vision to really reinvent photosynthesis or CO2 fixation, build a complete new to nature solution. It's almost like you would have a diesel car, a diesel engine, and then you would build an electrical car, an electrical engine, so a completely different design. But think about what you can also do in synthetic biology. You can, for instance, help to boost the diesel engine. You can make a turbo diesel. You can basically try to boost photosynthesis, not replacing it, but using the same principles and just try to improve photosynthesis and improve photorespiration. And this is something we can do already now. So what does it look like? That's, again, like a bit more technical. I'm sorry for that. This is how nature fixes carbon dioxide naturally, photosynthesis. It's the Calvin cycle. Rubisco is involved. And Rubisco fires, and Rubisco misfires. And it makes this compound called 2-phosphoglycolate, which is a photorespiration starting point. And this is toxic to the cell, needs to be detoxified, which requires a lot of energy, which takes many, many different steps, which releases ammonia, and more importantly, as I said before, which releases CO2 at large scale. And this is limiting photosynthesis to some extent. Now take again our thinking of a synthetic biologist.
think about this awfully complicated pathway that even loses CO2 on the way of mitigating photorespiration. How would I design that as a human being or as a synthetic biologist? I think we should not spend much energy to regenerate this molecule 2-phosphoglycolate. I think we should have a short pathway sequence, and I think we should also not release CO2. And this is what has inspired us to think about a new pathway in photorespiration that starts off converting 2-phosphoglycolate into glycolate, activate this compound through a CoA thioester, sorry for the chemistry. Then you actually fix CO2. You don't lose CO2, you fix CO2 to make a compound that's called tetracyl-CoA, and that's how we named the pathway, TACO, tetracyl-CoA pathway. You need 2 more steps, and you're back in the Calvin cycle. It's simply 4 steps, much shorter. It's probably the shortest possible sequence to regenerate 2-phosphoglycolate. And it needs just a minimum amount of energy. And this is shown here on the slide. How many steps, how many reducing equivalents, how many ATP you need. It's on the left lower side, just compared to natural photosynthesis. So it should be a super short and super energy efficient pathway. Very cool. The problem is each of these individual reactions does not exist in nature. And the most challenging one is actually this CO2 fixing step. It's an enzyme that we didn't know before. All right, so how do you go about that? What we do typically is we screen. We screen enzymes that can fix CO2, and we try to find an enzyme that shows a little bit of an activity, and this is shown here. So we want to carboxylate glycolyl-CoA, and we found a propyl-CoA carboxylase that could accept glycolyl-CoA. We solved the structure, and we looked at the active site. And that's a starting point for us. If you have the active site, you can shape the active site so that your real substrate fits much better in. So really you take it by the heart in synthetic biology by re-engineering the active site of the enzyme. And just to give an impression, I think with something about 0.01 turnovers per second. Okay, really almost nothing to measure. We had to measure overnight to see activity of the enzyme. But once you have the structure, once you start to redesign the enzyme, you can actually start to improve. It was around about probably 5, 6, 7, 8 mutations we had to introduce. So this is an old slide. We have probably tested around about 20,000 variants. We have a mixture of engineering by looking, rational design, and also just high throughput screens of random mutants. And what you see here is we actually improved the enzyme by around about a factor of 500, and we increased also its energetic efficiency and reduced it by about 30-fold. So what I'm telling you is you can actually create a new to nature enzyme that has parameters that are comparable to existing carboxylases. We did it also for the other enzymes. This was the biggest challenge, and we could put this pathway together, and we actually managed to get this pathway already into plants. This was with the help of Andreas Weber in Düsseldorf. And what we see right now is that these plants seem to grow better, especially under ambient CO2 fixation, where the photorespiration is a challenge. And they seem to grow a little bit better still under high CO2 concentrations. So we have an interesting phenotype. We still have to confirm if this is activity or if this is other stuff that's happened when you introduce it into plants. But at least that's one of the first examples where we have artificial pathway, a complete new to nature design, and you can even go inside of a more complex organism. I think that's the future for us, at least in the lab. All right, so that's almost the end of my presentation. I hope I could show you how we started off just by thinking about photorespiration, photosynthesis, and then we actually designed new solutions. We moved from the lab individual enzymes to pathways that we can operate in vitro, optimize with machine learning and automation, and then go into cellular environments, natural artificial cells, and in some cases even into chloroplasts. What we still miss are, of course, principles how to organize in three dimensions, in time and space, principles that, for instance, you also investigate in Dresden, like liquid phase separation or scaffolding. We also miss a lot of tools to turn on these pathways, regulatory elements. That's something also Thorsten is doing, but we probably also need in the future. We want to build complex designs, complex regulations, cascades, circuits. So all of this still we need to do, and we need to, of course, also optimize our ways how to bring stuff into the chloroplast. But we at least have ideas that there are solutions out there that it's worthwhile to develop these technologies. What I also told you is today that the climate crisis is a global challenge, and there will be not one single silver bullet. You need to think in different aspects of your life: transportation, energy, carbon capture. And for each of these individual parts, there will be individual different solutions. It won't be one carpet, it will be many different solutions. But I believe that SynBio also really has the potential to offer new solutions on the biology side, because we also need biology to address climate change. And we also need probably synthetic biology, because natural photosynthesis is not sufficient. I also told you that we actually use genetically modified organisms. So we have to talk about that. We have to go out and we have to talk to the public, and we need to discuss risks, but also benefits. You have to trade. You have to trade sometimes if you want to save the climate, or if you are afraid of genetic modified organisms. And we have a lot of experience. We have a lot of experience with GMOs, so there might be a solution, but of course we need to convince politicians, and we need to convince also the society that there is a safe technology maybe, or to be more precise, that each technology has its certain risks. AI also has its risk, a car has its risks. Even if I walk up the stairs, that's risk, right? You need to be able to balance those risks. You need to make fact-based decisions. And last but not least, I think it's also important to involve the younger generation. And what I'm really happy about is that some stuff that we have designed in the lab is already in certain textbooks. And this provides you with a great opportunity to provide, especially young school kids, with a vision that science can provide new solutions. As much as we were able to overcome the corona pandemic with, I would say, synthetic biology, some of the nucleotides were synthetic, right? So here is also new technology that we might actually be able to use. And the world is not necessarily gray. I think if we wrap ourselves together, we can show again that there is technology that can help us to overcome this challenge, which is a global challenge. All right. Last but not least, I think you're not politicians, I can quickly glance over it, but I think there's also a lot of potential in SynBio, and maybe it would be wise to think also strategically as a new field, as Thorsten just phrased it, chemistry, analytic, synthetic. The last century was the synthetic chemistry. It has brought a lot of wealth to Germany. Think about all the big companies, BASF, Bayer, right? What we have built on synthetic chemistry. Maybe we can also build things on synthetic biology. And the markets are there, and they will be there. And if it's only 1% of the predicted market, which is 450 billion euros, not too bad. All right. So that's the end of my talk. I would like to thank you again for inviting me. I hope I could elucidate a little bit. I hope I didn't disappoint you, that I didn't bring the super machine that saved the planet from global warming, but I could give you hopefully some inspiration that biology is cool to do, and that technical biology is also something very cool. Thank you so much for your attention. Of course, I have to thank a great team. I highlighted only a couple of people during my talk. It's a fantastic team, it's a national team, and it's a lot of different disciplines that come together in our lab. I'm of course grateful for the funding by the Max Planck Society, but also different funding sources, and of course my colleagues I've been collaborating with. I thank you for your attention, and I'm happy to answer questions. Thank you.
Okay. Yeah, thank you so much for this very exciting talk. So the future is open. Do you have questions? Yeah.
First of all, thank you for your talk. Very interesting and also motivating. I have a question about the workflow when you talked about the retrosynthesis, because I was wondering, how did you made up the reactions when you, after that, you searched for the enzymes? So how do you know that these reactions you created on scratch really exist? Because it's more work to do if you, you know what I mean, I think.
Right. So absolutely correct. So we are not just hovering in blank space. Of course, we have starting points. And I think the catch cycle, for instance, was built around CCR, and we know what reactions CCR or ECR does. So it takes crotonylco-A and it makes ethyl malonyl-CoA. So this was a block we knew this reaction existed. It was highly efficient. And then we build around the cycle. The TACO pathway is different. The TACO pathway starts off with a concrete metabolite, and you want to go back to something, and there you need to design a reaction sequence. So that is a different kind of design. So we have also ways how to look and design things, as I said. So either you start from a very good CO2 fixing reaction, and you build the cycle around it, or you start from a substrate and want to go to a product, and then you start to find the enzymes. In one case, it's easier, right? Catch cycle is easier because we have the CO2 fixing reaction at hand. TACO cycle has a different challenge, right? But you're absolutely right. That's the way we look at things. Mm-hmm.
So, yeah, Herr Kröger?
Okay, yeah, that works. You mentioned that enzyme that you can interface with electronic circuits. Is this something that only this enzyme can do? And if yes, why can it do that? And
Of course not. I mean, a lot of enzymes that do have iron sulfur cluster you can feed from the outside, right? You know this better than I do. So it's all about the chain of iron sulfur clusters inside of the protein, right? So you can, for instance, every protein has a ferredoxin, which is a small protein that can be loaded with iron and can accept electrons. It can do it. The challenge for us was here how to create an enzyme that can generate ATP, right? And so this is a specific enzyme because it can reduce an acid to an aldehyde and use very low potential electrons. And it has so much energy and power that you can actually create an aldehyde from an acid. If you go back, you can release one NADH, one reducing equivalent, and one ATP. So this is where we go. There's thousands of other enzymes that can simply use, for instance, electrons just to do a simple redox reaction. But here you need to create such a big electric energy difference that you are sufficient enough to make an ATP, which is a lot.
and the electrons come from?
Okay, so you can use, fair enough. So what we did in the beginning is we simply took, for instance, titanium citrate to feed the electrons from the outside, just to test if the cycle is operating. And then we adopted and put the protein on the electrode directly, and it can actually directly accept it. You can actually use the same principle to make and regenerate NADPH. So you can, for instance, take it's called FNR, ferredoxin NADPH reductase. You can also put this enzyme on an electrode. It will reduce NADH, and this NADH can then float around in the environment to actually be used by enzymes. So this kind of electrochemistry is prototyped. But I think challenge here was really to make ATP.
Would have another question, but
No, you can ask questions. It's fine.
Ask
My
another
train
question,
doesn't
so-
leave. So
okay. I'll quickly keep going. Completely different question. I mean, you alluded to that at the very end that structural organization is of course key in any system in a cell. So how much so far you don't really take care of it, it seems like, and do you think this is really something to invest in, and especially if you think about nature, does that many algae use a carbon concentrating mechanism where we really funnel CO2 directly to the rubisco to avoid this competitor oxygen.
fantastic question.
Is that something you're working on in this direction?
I'm not ignoring it. I'm just incapable of doing it. So this tells you how good synthetic biology is. I mean, so the point is what you see here. Just pulled up the slide here. Look at the chloroplast. You have this membrane stacks. You have then parts where the starch is stored. And one trick of cells is to separate chemistries which are not compatible of each other. And we have not used this principle necessarily yet. We have started to investigate, look at phase separation, for instance, to store some stuff inside of let's say, an own condensate to do certain chemistries there. We also think about maybe equipping the membranes we have with channels to let only certain things through. And really, if you think about a cell, what you said is correct. Think about a eukaryotic cell, a bit more complicated, right? So you do have the nucleus stores information. You have, of course, the mitochondria to make the ATP of the you have the chloroplast to fix the carbon. And it's also source sink. You can push a lot of more through if you create a source sink principle. So all of this is not implemented. And this is where I need the help of my colleagues, where I need the help of cell biologists, for instance, right, to tell me how to structure complexes or create metabolons, enzymes that have active sites facing each other's. And part of the success or not success of building systems is because we still have already too many chemistries interacting with each other. And I would love to actually build cages who would love to actually separate certain chemistries from each other. But this will take another 10, 15 years.
And maybe this is a direct transition to my question, which is, of course, I mean, what you achieved is massive, but you still, you're not in the cell really, right? So, I mean, what do you see as the challenges, or where I mean, you mentioned 2 or 3 already, but what are the challenges you're going for in terms of moving these reactions, moving these cycles really in whatever living system?
Yeah, that's a good question. So if I think about solution, I think in waves. The first wave is just boosting photosynthesis. I think that's doable, and I think we have good chance. The bigger challenge is to get these more complex systems up and running. In E. coli, certain modules work, but E. coli is not a chloroplast, and to jump from E. coli into a plant is also just crazy. So what we've been really working on is using unicellular algae and to build up a chloroplast toolbox. And I really think this is something that will be helpful, because you have a small microorganism, division times, short generation times, can multiply, paralyze. And then, of course, we need genetic regulation, right, to address and to express things in the chloroplast. So we have a basic toolbox now that we can use, Chlamydomonas, but it would be nice to have orthogonal parts, for instance, right, all of this regulation. And so it's, again, a mixture of building up technologies, multiply, paralyze, do all these transformations in the unicellular algae, maintaining this algae, but at the same time also having the genetic tools, right? So, you know, this is again like, why should it care about circuits if you don't have something you're aiming for to develop these complicated circuits? And why should I build metabolisms if I don't have any genetic regulation at hand? And I think this is where we meet again each other. And I think this is the next step, tools combined with pathways. And the more complicated it gets, the more complicated it will be. And I'm not sure, for instance, if orthogonality is important or not. We have seen this in cells. Orthogonality can be a liability, or it can be an asset. You never know what kind of effect the one thing has or doesn't have, right? So all of these things are super interesting for me, but also so hard to address. And this is also why we actually design so many cycles, because some of them will just not work in E. coli, or will just not work in the chloroplast, because we have overseen that one metabolite will not be functional, right? So this is why we also think about this more parallelization of pathways. Yeah.
So microalgae is currently your preferred direction, not like cyanobacteria to go to the bacterial photosynthetic organisms.
Yeah, currently it's clammy for us, and we have a little bit of cyanobacteria in the lab. But the point also is that we have, of course, a great environment. Felix Wilmund is now here in Marburg, who has a lot of expertise. So it also depends on your environment, what kind of organism you're working with. I mean, you know it better than I do, right? Yeah.
Yeah, thank you also from my side. A question from a former biologist, I might say. I used to be one, not a microbiologist. So when you're talking about like boosting photosynthesis, how about adopting or adapting photosynthesis? Because there was a recent paper in Nature, I'm not sure if you're aware, that in the canopy of the Amazon, it's becoming so hot that photosynthesis stops working. So, bold question, can you engineer something that photosynthesis can also work in like hotter conditions?
Well, microorganisms solve the challenge already, I think, because you have thermophilic algae, for instance, or microalgae, cyanobacteria. You know, it's a multifaceted thing. So, of course, it will happen automatically, but you know, the speed how this happens is probably a decisive factor, right? And you always trade something. So my answer would be for the wild. I mean, I don't dream of releasing organisms to the environment. This is probably not what's going to happen. But it's interesting for me to dive back into this challenge of what does it need to adopt, right, and studying it. And you can look at it and try to see how over 10, 20 years things will evolve, or you can try to do it in the lab. So as a matter of fact, we also touch again a little bit Rubisco for different reasons. We actually go back in time, because Rubisco is a very interesting historical angle, which is the enzyme started off, by the way, I need to get it back later on. It started off 1 billion years of evolution without any oxygen, and only then oxygen came. So now if you replay, if you re-awake and resurrect billion-year-old Rubiscos, which we have done now in the lab together with evolutionary biologists, and directly confront the enzyme with oxygen, maybe you can redirect the evolutionary past of it and way of it. And so these are things we are currently doing. And I think that's also, again, like it's a mixture of synbio, but also looking back into what biology has solved and which path it has taken. It's more like, very often you think about biology being creative, finding new solutions, but the older a thing gets, the more complex it is and the harder it gets to go back in time. It's like bureaucracy at universities. So they are just too complex and they tend to grow and to become even more complex because every person has a certain function in there. And I think that's what you see if you see a protein which is 2 billion years old. It's hard to go back without losing function. There are certain interconnections which are hard to do. And so you learn something if you break it down into easier parts and you evolve it again. I think that would be one solution too. But again, it doesn't help right now. But it gives an idea of what the landscape is and what paths are blocked or open. Yeah.
Thank you.
Yeah, another question there.
Have you only focused on creating artificial pathway cycles? And if you do so, what is, again, the advantage of only focusing on cycles and not on a linear pathway, creating an artificial linear pathway?
Yeah, that's a very good question. So the point is that you need harder chemistries to build carbon molecules. If you have a cycle is as follows. Well, actually, I can use a blackboard. It's cool. I always wanted to do it. Here's chalk. Fantastic. I always wanted to do that. You know, if you get carbon in, for that matter, you take carbon is always red for me. You throw it onto a compound to make a CC bond, okay? And then you kind of reduce it along, and then you can take the fixed carbon out. So that's a chemistry where you make a CC bond first, and then it's easier to handle this molecule. If you think about linear pathways, you need to reduce a CO2 molecule, which is much harder and very often requires anaerobic conditions. Not good here in this atmosphere, okay? So this is why we actually think about cycles, about these carbon cycles, because it is much more compatible with transformation of CO2 in an easy way. Otherwise, you can, of course, reduce, right? Then you get it up here, but that is problematic under CO2 conditions. And you still need to make a CC bond formation. You do them later on. But in principle, it's about this anaerobic and the investment of the energy you have to do. Does it make sense? Mm-hmm.
So maybe I ask the last questions.
Sure.
How about computational modeling? Would this help speed up your experiments just to predict or build like a digital twin of a chloroplast?
That is also a very good question. So I want to answer in 2 or 3 different layers. Even pathway design now can be automated. So there is tools that just browse through all the enzymes listed, 40,000 different enzymes, and there's actually automated pathway generation software. This one lacks still the enzymes that you think of could bridge certain metabolites. Okay, wow, party starts. Second point is digital twins, perfect, but what we, for instance, lack still is a great understanding of concentrations. You know, compartmentalization, just as we talked about. If you break a cell, you get, let's say, 0.5 millimolar CoA all across, but within the chloroplast, it's probably, you know, so we actually lack ways to really get the information we need as biochemists. And that's again, and maybe the last important thing, we think also about enzymes doing transformations A to B, but of course, enzymes are also taught and trained to not do other reactions. And typically, enzymologists tend to measure the one reaction they want to measure, but all the other things not. And this also makes it hard to predict how a system will react. I think this is that we have to face. A digital twin would be perfect. The cool thing in biology is you can model a lot if you take the right parameters, but if it's then predictive, very often not. Thank you so much.
Last question,
Yeah?
do condensates play a role here that compartmentalizes?
Yeah, so that is something we are keen on doing. We actually also played around a little bit with doing that. And I think that will be part of the solution will be also to have very interesting condensates as a way to structure certain parts of metabolism or to concentrate certain metabolites. We have already tapped a bit into Rubisco and condensates, right? And we actually see that they speed up, for instance, CO2 fixation. So there is cool things that we would like to explore in the future. Maybe with Dresden, I'm not sure. Yeah.
Okay. Right. There are obviously no further questions. Thank you very much again for this exciting talk and inspiring talk. And we hope we will see you again here. Thank you.
Thank you.